Okay, so we're at 30 minutes later, and we have our test tubes, remember, that have 0.1%, 0.01%, 0.001%, 0.0001% bleach. All of them have been inoculated with that E. coli, and then we have our control, which is our water. So what I've done is I've gone through and I've actually created the plates, already labeled them. And so what we want to test now is we let the E. coli sit in there, let it multiply, let it get introduced to that bleach. So we want to see if any of the bacteria that is now in that bleach solution is going to grow. So what we're going to do is we're going to swab each one of these plates with the appropriate treatment. So for example, this first one is that 0.1%. My plate's 0.1%, so that's the plate I'm going to use for that first tube. And what we want to do is we want to take these sterilized cotton swabs, and we're going to open up. Before we open up, we're going to make sure we shake that pretty well. Want to open up that tube, and then we want to make sure that we get that cotton swab nice and wet. Once we do that, we're then just going to basically streak across the entire plate. Make sure you get the entire plate. Kind of move that plate around. Do multiple streaks, just to make sure that we get that on there so that way we can actually see the growth. So now that we've used this first one, I'm going to put in the back so that way I know this one's been inoculated. And then I want to take an autoclave bag and stick that used cotton swab in there. So our next treatment was that 0.01%. So once again, we're going to shake that tube up. Once we're done shaking up the tube, we can uncap it, grab another one of our cotton swabs. Make sure we fully submerge the cotton swab in the tube, and then we're going to do another streak. Done with that tube, move it to the back. Third one is that 0.001% treatment. Same thing. I'm gonna mix that up. Grab a new cotton swab. And then once again, streak. Making sure to get all over the plate. Our last bleach treatment was the 0.0001%. So once again, shake that one up. Grab a sample of it, and then do a nice swab. The last one we're doing, once again, is our control. This one has no bleach, so the whole point of this one is we want to just see what kind of growth we would get by just placing that E. coli in water. So the treatment is water, but it's just kind of our control because we have no bleach whatsoever in this test tube. Once we are done with all of these, you'll notice I'll actually put my initials on it, put the percentage on the back as well as on the top of the plate just in case. But we're going to put these all together as a group, and then once again we're going to stick them in the incubator for 24 hours in the inverted position. Remember the inverted position because you want to make sure that you don't have condensation on your agar, but you want it on the lid instead. So we're going to let that sit for 24 hours in the incubator, and then I'm going to get pictures and post the pictures so you guys can see the results. Once again, we're trying to test what concentration of bleach is going to be most effective at killing E. coli.